Welcome to Your Amazing Brain Live. We are here at the University of Oxford to record a live experiment here that we're carrying out right now. So I'm Dr. Stuart Clare. I'm a scientist who uses MRI scanning to take pictures of the brain.
And my name is Dr. Miriam Kleinflügge. I'm a neuroscientist. I also use MRI scanners that we're going to look at today for my research, and I usually look at how we make decisions and what happens in the brain when we do so.
So this afternoon we're going to take you on a tour of the facilities that we've got here, and we're going to then run an experiment live that's been suggested by one of you that's watching. We're going to then look at the results, see what they tell us, and hopefully plenty of time for questions that you can ask.
And I think on the screen right now, you will see how you can ask your questions. So get your thinking hats on, try and think of questions as we go along. You can type them in. We'll try and come back to them at the end, but we're really curious to hear your questions. And when we visited some of you in the classrooms, you had absolutely amazing questions. So we look forward to that. I think I probably don't even need to remind you, because you're super familiar from your school with how we do scientific investigations. But in case you want to take notes, or maybe even have your brain experiment report in front of you, you can jot down things as we go along. So I'll just remind you briefly of the steps. So normally what we do first is we ask a question, and that's actually the job that you have done for us. So we don't need to think of the question. We will introduce the question that you asked for us today, and we make predictions, which also you have done in the classroom. At least some of you who have put entries in really well. And so that's the second part of the investigation. And then the third part, that's where we're coming to today. We're setting up the test, and then fourth, taking measurements. We're observing and measuring, in our case today, what happens in the brain when someone does, well, we won't reveal it quite yet, but we will in a minute, which experiment we're actually doing. And then while we record the data, you can write down what you're seeing coming in, and at the very end we will be interpreting it, seeing what it means, and we might be doing some evaluation. And often what we do in science is to then come up with new experiments that we can test, but today we'll stick to just one. So before we start, we'll introduce you to how we take our measurements today, and so you will see a tour of our MRI facilities now.
So we're standing inside the control room of the MRI scanner at Oxford University. And inside there is the MRI scanner. Let's take a look inside. So MRI stands for magnetic resonance imaging, and it uses a really strong magnet. This is not like a magnet that you just find on your fridge. This is 12 tons of magnet. It is incredibly strong. It could pick up a car. Let's just see what would happen if we took something that was magnetic into the room. So you can see here, in this plastic bottle, I've got a penny coin. Let's see what happens when I take it inside. So you can hear it's quite noisy in here. There's a pump on top of the magnet that's keeping it cool. But if I take the penny coin close to the magnet, you can see how strong the pull is on that coin. In fact, if it wasn't inside that big plastic bottle and held by the string to my wrist, then we'd probably lose that coin and never get it off the side. So that shows you how strong the magnet is. Let's put this safely out of the way for now. So, what we do to take pictures of someone's brain is we ask them to lay on this table here. They put their head inside this detector, which is really close fitting, which gives us the maximum sensitivity to the signals inside the brain. We get them to lay down, make them very comfortable, and then we put them right into the centre of the magnet, where it's strongest and most uniform. So we can start taking pictures inside their brain. So there you've seen the equipment that we're going to use today.
But what about the experiment? Well, devising the experiment has been part of a wider project that we've been doing with schools in the Banbury area. Perhaps you even visited the Your Amazing Brain exhibition in Banbury, and you saw the exhibition and you were thinking about ideas about an experiment that you wanted to do on our scanner here. So you've asked the questions and made predictions. So we're going to come together and record the data and make the measurements and record the data. So what were the ideas, Miriam? They were really good, weren't they?
Yes, so we had some really fantastic ideas. So we want to outline some of them, even if they're not the winning entries. So here are some that several of you suggested. For example, quite a few groups thought it would be interesting to look at different tastes. How does the brain react when you taste something that you really don't like, something that you find disgusting, versus something that you find really nice, you know, your favorite ice cream, chocolate, something. So the question is, why did we not pick it as the winning entry? We think it's a really fascinating question, but actually it's not really practical to do it. It would be really hard to have our participant in the scanner eat an ice cream. And that's, as you saw, because it's a very narrow tube and the head is in this head coil for us to do our measurements. So it will be really hard to give them something to enjoy. And what we do sometimes in experiments, and it's really tricky, is to give them liquid through a tube. But you can imagine it's not as nice and it's also quite hard to actually do. There's another problem with when you chew on something. Imagine what happens when you chew on something. It actually makes your entire head move to some extent, your jaws move. And so you can imagine that our pictures would actually get blurry. So while we liked the ideas, and this was from the Brain Explorers team, the Brainies team, and the Scientist Eagles teams, we really liked them, but we can't do them, unfortunately. But keep thinking.
Okay, so coming on to the runners-up. So we've got two runners-up. And the first runner-up was the Big Brainers. Now, the Big Brainers asked the question. Now, interesting, in their team, they had people who spoke two languages. They'd grown up speaking two languages, and they wanted to know why they found it so easy to switch between speaking each of those languages. And that's a really interesting idea. And we do know from some of the scientists who've studied language that if you grew up in a family where you were speaking two languages from birth, and that was always heard those two languages, then the same part of the brain is working whichever language you speak. But if you learn a language later in life, then it's a different part of the brain that you use to learn that language later in life. We thought it was a really brilliant idea and would have loved to have done that experiment. We just didn't have someone who spoke the two languages from birth, so we couldn't do that one.
And then we're coming to our next runner-up. So we also really like this idea. It was from a team called MSAVGG. I think that's the first letters of your first names, potentially. And so your idea was to look at kids who live with different kinds of experiences. So you can imagine some kids are always schooled from home, and some kids actually go to school every morning, as many of you do. You do, I think. It's where you are right now. And so you wondered what is different about the brains of kids who go to school. And we have all experienced homeschool is quite different in lockdown. And so that would be a fascinating experiment, but we would actually need two participants at least to test this idea. Someone who does only homeschooling, someone who does go to school every day. And so again, that's not really practical, but a really brilliant idea and really interesting.
It was a really great idea. I really like the way that you'd obviously thought about your current experiences and was wondering what was going on in the brain when they had that. Brilliant idea. But that takes us to our winner. Drum roll, everyone. Oh. So the winner is from the Exquisite Emotions team. They're at Harriers Academy in Banbury, and they came up with a brilliant idea, and we're going to give it a go. So they wanted to know which parts of the brain were active and busy when the people were afraid of something, but also when they were comforted by something. And so they wondered whether same areas or different parts of the brain were active when you were afraid and comforted. And we thought that was such an interesting thing to look at. And so that's what we tried to do. So Miriam, how have we put this together to make an experiment
so to make an actual experiment from that, we have a participant lying in the scanner right now for us. Her name is Barbara. You'll get to see Barbara in a moment. And Barbara, basically, we talked to her and we asked what sort of thing would make her feel afraid. And Barbara really doesn't like the animal octopus. She just doesn't like looking at octopuses or thinking about them, and it makes her really anxious and afraid. And so we thought one really simple experiment we could do with Barbara then is to show her pictures of different octopuses, and you will see one of the pictures that she will get to see in the scanner on the screen now. And so basically we will show her a series of these kinds of pictures and ask her to imagine you know, seen these octopuses in real life. Of course, we can't make it real here. It's just going to be pictures, and make her feel a little bit scared. We don't want her to be too afraid, because that's not nice to do to your participant, to your volunteer. But we do want her to be a little bit anxious and see what happens in the brain. But then we also need to compare it to something. We want to have a situation where Barbara is not afraid. And so we asked her what kind of picture would make her feel very comfortable or at peace. And she said that she really likes little kittens or baby animals. And so here is one of the other kinds of pictures that she will see of a kitten. I'm sure you all love looking at those as well, at least I do. And so for a second part of the experiment, then we will ask her to look at kittens and feel really comfortable in the scanner. And so what we want to know is, what would you have picked? So we have a little poll for you that you can fill in if you like. And so what we're asking you to put down is, what would make you feel afraid? It might not be an octopus like for Barbara, so feel free to type in your questions, your answers, not your question. The question is, what would make you feel afraid? You have 1 minute to do that. Okay, well, thank you so much for participating, for those who were able to. As we can see, people are afraid of all sorts of different things, and that's quite normal. We aren't all very different. So some of you really don't like the dark, some of you really don't like slugs, most of you do not mind octopuses. But actually, Barbara is right there in the scanner, and we want to get going with the experiment. So I will just check in with her. That's something we actually do when we run experiments, to make sure our participants are happy. So I will just talk to Barbara. Barbara, can you hear me?
Yeah, I can hear you.
Perfect. Are you doing okay in the scanner there?
Yeah, I'm good.
Can you just confirm that you are really scared of octopuses for the children who are listening?
Yeah, I hate them. I hate octopuses.
Okay, so we know now Barbara really doesn't like them. So Barbara, we're gonna go ahead with the experiment. Just a quick reminder to really try and feel afraid of these octopuses, even if they're not quite real. Try your best to be a little bit scared. Does that sound good?
Yeah.
Okay, here we go.
Okay, so here we go. Okay.
So the measurements are coming in now as we speak, basically, but it takes a little bit of time. So Stuart will talk us through what we're going to see.
So right now we're taking a movie of what's going inside Barbara's brain. But before we look at those movie pictures, we want to look at what her brain looks like. So just a bit earlier, before we came on the live experiment, we took a picture of Barbara's brain, and I'm just loading it up onto the screen now. So let me take this model. I've got a model here, which is a 3D model of a brain. And you can see here are 3 photos inside Barbara's brain. They're MRI scans, and this one here is imagining that we're cutting the brain in this direction here, okay? And then this one here is imagining we're cutting through the brain in this direction here. And this one here is cutting in this direction there. So you can see, we can see all through the brain, and actually I can move up and down through the brain, seeing every different slice through the brain. Now, in Barbara's brain here, the grey area around the edge of the brain here, they're the brain cells or neurons, and then all of the connections in between, which are showing up bright white on the screen here, they are the connections, the fibres that connect different parts of the brain together and are really important for how the brain works. and that's called the white matter. So that's a bit about what the brain is looking like, but actually to know which parts of the brain are working hard and are reacting to that fear from looking at the octopus, we need to look at something else. So yeah, explain that, Miriam.
Yes, so right now, we're taking a different kind of measurement. So this is a static picture of the structure of the brain we were just looking at, but actually what we're interested in is where does the brain activity happen? And that's really hard to look at directly, but what we know is that wherever the brain is active, we need a lot of fresh blood because there's oxygen needed. And so basically what we're measuring here with our MR scanner today is where the blood flows in the brain. And so one hypothesis that we can already make is that Barbara is looking at pictures of the brain. So one part of the cortex that cares about looking at pictures is the visual cortex, which takes in all the things that we see with our eyes. So in this brain model here, you could imagine the eyes being at the very front of the brain, but actually the visual cortex is all the way at the back, which is a little bit intriguing, isn't it? But there are these like huge nerve fibers, which an optic nerve, which transmits the signal that comes in through the eyes to the back of the brain, to the visual cortex. And actually it's the left hemifield that we process with the right part of our visual cortex. So the information crosses. And so since the pictures are in front of Barbara on the screen, we would expect both the left and the right visual cortex to care about these pictures. And so that's something we can check now in the measurements that we're looking at. So what Stuart is showing here on the screen is now a color on top of the structure of Barbara's brain. And so you can see in some red and yellow colors that here at the back of the brain in the visual cortex, there's some activity which is happening right now live as she's looking at the picture of the octopus.
So okay, that's what she's looking at, the activity she's looking at. But is that about her being afraid of octopuses?
Yeah, so that's a really good question. So even if there were any other pictures that she was looking at, we would always see the visual cortex because we just know it processes the visual information that the brain is taking in. But there will be some other areas that we could check that I will try and explain to you as we go along that might care more specifically about the fact that she's actually afraid of the pictures that she's looking at. But we won't know for sure from just looking at these images at the moment if there are regions of the brain that are specifically caring about the fear in the stimuli, and that's because we haven't anything to compare it to. But maybe we can just have a look at what other areas we find, and then later check when we're looking at kittens if we see the same brain areas or different brain areas. So if I just take a scroll through, so what I'm doing here now is I'm scrolling from the back of the brain where we know we should see a lot of activation in the visual part, and then I'm scrolling further forward, and we know that more complex visual information is processed in infratemporal cortex, which we can see here. So that's still part of visual cortex. There's actually quite a few different parts that all belong to our visual processing hierarchy. And then let's see what else we can see. So there's actually here some sensory motor activation. So that is part of the brain that makes us plan and think about the movements and also the things that we might sense. So she might have some imagination about how it would feel to touch an octopus, for example, or what might want her to run away from the octopus. So that could be why, but of course we're moving into the interpretation now. So maybe for now we should stick to just observing. So we can definitely observe some activation here, which is more in sensory motor cortex. We see some activation further down, deep inside the brain, which is in the brainstem. And then this massive visual cortex activation. So that's, I think, the observations that you can be writing down as the measurements are coming in.
Great. So really we need to compare these not just with the pictures we've done now, but we need to compare it with her, something she's comforted by.
That's right.
So should we run on to that experiment next?
Yes, and actually as we get that second part of the experiment with the kitten started, we have a question for you. That's our second poll, and we want to know, will the brain show different activity? What do you think, true or false? Will the brain show different activity when we see something scary versus when we see something that makes us feel comfortable? So can you type in if you think that question is true or false? Will it look the same or will it look different? Which is basically the experiment that the winning entry suggested. So that's what we're coming on to now.
Great. So what did you predict there? See whether there's any results coming through. So I think most of you thought that there would be something different when it was scary compared to being comfortable. So let's have a look and see what's happening. So we're now just starting that experiment, and this time we're showing pictures of kittens and some other cute animals. So hopefully that's a much more pleasurable experience for Barbara in the scanner. So we shall see. I'm going to just, there's a couple of things. With any live experiment, there's always things that you have to check that it's all running. I think we're okay. Yes, that's started to run now.
So obviously it will take a little moment for those pictures to be processed and to come through. And maybe in the meantime, we can talk about our predictions. So we know that some parts of the brain really care about when we get some positive stimulus, when we get, you know, when you think about ice cream, or when you think about something, your favorite holiday, or something else, someone you really love. And so those are the reward processing parts of the brain. And so one of them is really deep inside the brain called the striatum, which many of you may not have come across yet, but maybe in a moment when the pictures come in, we can check if we see some reward system activation for when Barbara looks at something that she finds a lot more rewarding, a lot more fun to look at, such as these kittens. And just now before, when we looked at the fear, there was nothing in the ventral striatum, so it wasn't something I could highlight to you because it was just empty. So that's something we can check for in a moment when the pictures come through.
Okay, so these experiments are taking a little while to do. Why is it taking a long while to get this information? Why don't we see it straight away?
Yeah, so there's multiple reasons for that. But for example, the blood actually takes a few seconds to go to the brain areas that are active. So even if there's a picture on the screen right now, we only actually see the activation about 5 seconds later. And then often it's not quite strong enough to see it from just one picture, but we show multiple pictures for a long time in a row so that we really have enough power to see the results on the screen. And actually it turns out that normally when we run our experiments here in the lab, one participant is also not enough. We want to be really, really sure that our conclusions hold in general and not just for one specific participant. And so while this is obviously really great to do live, normally we would do another 29 sessions, say, to get 30 participants all in all, or maybe even something like 50 participants all in all. So we would be repeatedly doing the process that we're going through today and then check that it's similar across all participants.
So I can see we've got that visual activation again, which is good. So maybe if I can try and move back a bit. Oops. Move that back a bit. So we're getting the visual again, which is not surprising because she's looking at pictures again still, so we would expect to see that. But what about those other areas? What about that ventral striatum area you talked about? Are you seeing anything there? Let you scroll and find? Not yet.
I think it's probably a little bit weak still, but I will tilt it a little bit in a moment. I'm just sort of adjusting the color so that we can see things a little bit more clearly. So yeah, here we go. There's more things coming up now. And actually, so you can imagine what I'm doing here is I'm scrolling, as Stuart has just said, from the back, kind of further forward to, if I go all the way to the front, we would be here at the level of the eyes, just to orient you here. And so here we're in the frontal cortex. And then as I scroll through those older parts of the brain that really care about reward, they're actually very deep down inside the brain. So this, for example, could well be a region that has a lot of dopamine. It's one of those like neurotransmitters that lots of people think about, one of those brain chemicals that lots of people think about when they think about reward. And so you can see actually this activation wasn't there earlier, but it is now coming up when Barbara is looking at kittens, which is really intriguing. And you can see there's a really old structure here, which you can see in gray, both here on the left, I'll just show it with my finger, and here on the right, and you might have come across it. It's called the amygdala, and a lot of people think that the amygdala processes emotions, so not just fear but also happy things, but in particular it really, really helps us to run away from something that we're really afraid of and to kind of activate this fight and flight response. And so you can see there's not that much activation in the amygdala here when Barbara is thinking about kittens and looking at kittens, because obviously she isn't keen to run away from them. In fact, she would probably want to stroke them and approach them, and there's no fear involved. So that's a really interesting distinction. I'm just trying to kind of scroll through and see if there's anything else interesting that I can explain.
And while you're looking at that, I'm just going to have a quick look at the questions. So if you've been asking questions, and hopefully they'll come up here, and we have got some questions here. Oh, there's a really interesting one. I wonder if we could just answer that one now while you're looking there.
Yeah.
What does your brain do when you sleep?
Yeah, it's a really good question. So sleep is incredibly important. So please don't cut down on your sleep, but sleep enough. Because the brain really needs that rest. So there's some neurochemicals that build up as we go through our day and that we need to clean up. And that's actually one of the things that happens in our sleep. The brain kind of cleans itself up. And it's really, really important that we don't skip that part, because then it's fresh in the morning and it's ready to go through the daily processing again. But there is actually also some brain activation while we sleep. And so that's, you can imagine it could be related to the dreams that you dream, which are actually something we don't understand that well. But there's also brain activation related to memory when we sleep. And so we know that some of the experiences that you have gone through during the day will actually be replayed while you sleep. And that strengthens the synapses in your brain and makes us remember them for longer.
Brilliant.
Yeah, so that's a really brilliant question.
Sleep is so important, isn't it? You just don't realise it really can affect how much, not just feeling tired, but actually how you learn and how you develop.
So we have actually just finished this experiment, so I'll just check in that Barbara is doing okay. Barbara, are you doing all right? Oh, I did. Barbara, are you doing all right?
Yeah.
Perfect. Okay, well, I think maybe it is worth remembering that there was one other thing that the winning team was actually interested in. I forget the name of the winning team, the Exquisite Emotions team. And actually, we really, really liked the idea, partly because of what you put down about how can we make ourselves feel more comfortable when we are afraid. And so far, we've really just looked at when Barbara is afraid and when she's comfortable. But there is a lot of scientists interested in this question. How can we make people less anxious when they are anxious? Because we all sometimes are anxious, right? And so one thing that the Exquisite Emotions team actually suggested is, can we make people less afraid, even when they're looking at something that they're afraid of? So one last thing we thought we could do to test if that's possible is to give Barbara octopus pictures to look at, but ask her, instead of being afraid of them, to think of something that makes her really comfortable. So to think of kittens while she's looking at the octopus pictures. And I must say, it's probably an experiment that might not work live, because it's a very, very subtle question. And it's one of those where maybe we will actually need 30 or 50 participants to look at. But why don't we give it a go?
Yeah, I think so.
We should just give it a go. But we don't know what we will find.
Let's go. So
I will let Barbara know.
Yeah.
Okay, Barbara. So for this last part of the experiment, we will ask you to look at octopus pictures, but actually imagining stroking kittens, being around kittens, feeling really comfortable and being really peaceful. Do you think you can do that?
Yeah, sounds good.
Okay, here we go then.
Okay, so we'll start the scanner off again. So you can hear, maybe you can hear a few noises while the scanner is going. I don't know if you can pick it up on the microphones, but the scanner is quite noisy, and that's something that we just can't avoid. It's part of taking the pictures. But what we've done is we've given Barbara some earplugs that go in her ears to make it much more comfortable for her. So it's not too bad. And we've both been in scanners before, and it's not too uncomfortable. We'll try and make people feel as relaxed and comfortable as they possibly can. So we'll let this run. I suggest we let this just run and maybe answer some questions, because I think we're not going to see, if we see any results at all, they'll only be right at the end when we've got a lot of
Enough
data.
Pictures. That's right.
But let's head back to the questions on here. So we have got more questions. Do you want to pick a question from there?
Okay. There's some really good ones, some really big ones. So,
Yes.
does the brain glow in the dark? No. That's a simple one. We can just answer that with a no. Do parts of the brain light up when you eat or drink? Yeah, so that was basically one of the taste questions, really, that one of you suggested. And yes, there are definitely some areas of the brain that make you move your jaw, for example. So any movement that your body does is steered by your brain. So you will definitely see some brain activation when someone eats or drinks. And maybe it goes beyond just moving your jaw. It might also be rewarding when you eat or drink something. So there will be some of those striatal areas deep in the brain, for example, involved.
Yeah. I think there's an interesting one again, linking into the fear thing. Was, why are we afraid of things that cannot really hurt us, like tiny spiders and roller coasters. And that's really interesting because we know that some of our fears really aren't these things aren't going to hurt us. They're not going to. There's no real threat. We know that roller coasters are safe. We know that tiny spiders, certainly the ones in this country, are not going to cause any harm at all. So why are we afraid of them? And I guess partly that is because we've learnt an association over our lives, but also from how others around us respond to different things that are exciting and not. And I guess there's also some, all over evolution as well, that we have learnt to be afraid of some things that maybe were scary to us. So the fact that spiders are scary possibly comes from times when that was really a matter of life or death. And we've sort of got this hang-on idea that still these things are still scary right now.
Yeah, and I think probably humans didn't evolve around roller coasters, and previously probably it wasn't a good idea to go high speed down like a really steep slope, because normally that would result in death. So I think it's maybe okay that our brains are scared of that thing. But we have some really sort of advanced part of our cortex that we particularly have as humans or non-human primates, so that evolved later, like the prefrontal cortex, for example, and that can really help us to kind of regulate our emotions. So that can then help us to say, well, but it is only a roller coaster, even though normally you should be afraid going down at high speed a hill. Here it is actually safe in this context. So that's where sort of the older parts and the newer parts of the brain interact with each other to regulate emotions. And it's something you can practice at home. And when you're somewhere where you're scared, you can really try and practice thinking of something positive. And that's a strategy many people use, and that really works.
Yeah. We've got a question here, which again links a little bit to some of the stuff we've seen today. Which part of the brain activates when you experience pain? And I think it's probably true to say, and I'm not a trained neuroscientist, you've done this more than I have, but I think probably some of these same areas we talked about today would actually activate really strongly if we were causing pain. So there are some experiments that some people do, where with the permission of the person, they give them some pain. Maybe it's a little pinprick on the skin or something there that's just is a bit painful. And I think if we were doing that in the scanner right now, we would see exactly those same areas really lighting up very, very strongly, because our brain is really well tuned to responding to pain.
Yeah. And I wonder if some of you are noting down the observations that we're making as we go along. So remember, we're kind of in the process of recording data and observing and measuring. So, the middle sort of fourth and fifth part of the process of the scientific investigation before we get to the interpretation. And so before we look at the results from this experiment, and I truly don't know what they will show, the sort of things that we might be looking at, particularly when we observe and measure now, are, do we find the amygdala, which we think responds more strongly to fear than to comfort, but it's now something in between. We're looking at fearful stimuli here. Barbara is looking at the octopus again, but she is trying to feel comfortable. So do we see the amygdala or not? I'm not sure, but that's something we can look at. And then what about the ventral striatum, that region that really has a lot of dopamine and, the happiness hormone, if you like, although that's a simplification. So shall we have a look to see what we see in the striatum and what we see?
So that's the old
in the
results. So I need to load up some results that we're just collecting now. Okay, so we've just collected all of the data and go down. We're
just loading it in now.
There we go. And I think probably what I need to do, sorry. I'm going to drag that onto the screen there. Sorry. And there we go. Okay, so here we go. So look, I'll give you the control there. Yeah, do you want to? Yeah, I'm going to turn. So we make a few changes to what we're seeing to make it clearer to see. So we'll do that. Oops.
So while Stuart is adjusting what we're seeing, I can also answer one other question that was on the chat. What about the size of our brains? Everyone always asks that. Are people with a bigger brain cleverer? And the answer is no. There's no correlation between brain size. And that's good news for all the females in the audience and me, because generally female brains are a little bit smaller, but it does not mean that they are more or less clever. People differ in how clever they are, but it's not something that relates to their brain size at all.
Right, I'll let you.
Yes, I will have a look at these images that have just come in. And I will try and point out the things that we see again to you. So just as a reminder for those who are noting down their measurements, here we have the visual cortex again. Not surprisingly, Barbara was still looking at pictures, and so we would expect there to be visual cortex activity. You'll know really well now, I think, where the visual cortex is. This is still part of the visual processing hierarchy in inferotemporal cortex, so it's part of the temporal lobe of our brain. And so now we're coming to the more interesting bits. I might have to just double check what we're looking at here. So do we see some striatal activity? So the striatum is kind of just here where we're looking, and you can see there is a little bit in the striatum. So I think probably that means, in terms of interpretation, Barbara did manage to make herself feel a little bit more comforted, or maybe the thinking about kittens made her feel a little bit more rewarding, more happy. I cannot see any activation in the amygdala. So again, the amygdala is a little bit hard to find, but it's this gray circle on either side here in the left and right hemisphere, and there isn't a lot of amygdala activation. So it's always really important to be careful when you see a result that just didn't come up to say it's definitely not there. But at least the first hint here is that this is a little bit different to when she was looking at the octopus, where we expect a lot of a big fear response in the amygdala. So maybe it did actually help for her to like be less anxious looking at octopus images and thinking about kittens. And maybe we can even ask her briefly what she thought. Barbara, we just looked at these images, and I wondered if you managed to make yourself feel more comfortable looking at these octopus images, thinking about kittens. How did you feel?
Yeah, it felt better than the first time, but sometimes it was still scary to, like, when some of the pictures kind of jumped out.
Okay, thank you. Can you just repeat that, because we couldn't quite hear it as well? Okay, so I will repeat it because that might come through for you in the live stream a little bit better. So what Barbara was saying is that it did feel less scary than the first time around when she looked at the octopus images, now that she was thinking of kittens, but it still was a little bit scary sometimes. So I think that's quite consistent with the data that we see here. And I think that a lot of people also who work on these things say that it takes practice to regulate your emotions. So she would maybe need to practice it over and over again when looking at octopus images to think of something that makes her feel comfortable. But I think that's, a success, and that's congratulations to our winning team, really, because the Exquisite Emotions team came up with a fantastic experiment here. And I think, we could, of course, run that in many more people and be a little bit more confident of what we see. But I think it's a really encouraging result. And also to hear from Barbara that it actually made her feel better to think of kittens. So I think that's quite exciting.
It's really good, really great. It's nice to see the experiment work so well. Have we got time for a few more questions? Yeah, we've got time for a few more questions. So let's pick out one of those. Oh, why do I find maths difficult? That's an interesting one now. We're not thinking back to when I was at school and thinking about maths, and it sometimes just feels so hard, doesn't it? But I wonder whether one of the things we know about our brains, and this is true whatever age you are, so even if you're a teacher watching here or you're at school right now, that the more times you practice it and the repeating, not only it's not just you're used to doing it, but actually it changes your brain. It makes the connections in the brain stronger if you repeat it again and again and again. So anything that you find really difficult, so maybe it's maths, maybe it's, I don't know, you find football really difficult, or there are other things you do. If you keep practicing, then actually your brain will strengthen and connect and connect and connect and do better. So maybe you think, well, why am I not getting maths results like my friend who could just do it just really easily? Well, if you keep practicing at it, keep doing the same again and again, your connections will increase, they'll strengthen, and that will make you faster and quicker and better. And so perhaps don't give up at maths just because you find it a bit hard now. Keep trying, and you will learn it. It will get better. And we've done brain scanning experiments just like this, where we've looked at that and we've been able to measure the differences in children's brains as they learn maths, and we know that that is what's happening. There are changes that go on, and so stick with it, keep learning maths, and you'll get there.
Yeah, perfect. I wonder if for those of you taking notes, we should briefly recap what our conclusions are from the experiment. So there are some structures in the brain that really care about reward, and they were the ones that we could see when Barbara was looking at kittens. And so, for example, the dopaminergic regions really deep in the brain, or if you want to write the name down, it's the ventral striatum we saw. But then there are some other regions in the brain that process when we are afraid of something. And that's a good thing sometimes, because sometimes when we're afraid of something, we should run away. And for example, we focused on the amygdala there, which is a nucleus deep inside the brain. So if you want to write that down, this is sort of our evaluation of the study. And we saw that there was less activity in the amygdala and still some activity in the ventral striatum, our reward region, even when Barbara was looking at the octopus, but thinking about kittens. So there's ways we can regulate our brain activity to make us feel more comfortable, and that could help us. So I think that is sort of what the experiment came to in terms of the conclusion.
That's brilliant. It's really exciting to see a live experiment. It's something that we've put together, a brilliant idea from the children who sorted that out. So really well done. And,
should we see if there's any other questions in the chat? So feel free to type your questions down as we speak, and we will keep...
Some of these we've answered, but let me see what others we haven't answered here. Some of these are so big questions.
There's some really great ones on how our senses work, and I think we should do, we could do a whole extra lesson on just the visual system, on just the system that helps us to feel things with touch or to make us move. And they're all really fantastic questions. And so I think, keep learning. And there's a really great book that a friend of mine published, which has lots of explanations on these things, and you can find it in the bookstores.
Okay, how about this one about brain reacting in different ways when you speak different languages? And we sort of covered that a little bit earlier with that other experiment about languages, and I think it's really interesting. And actually, when that experiment was suggested, I spoke to some of the colleagues of mine who actually researched language, and they said to me that the big thing about that experiment would be when you learnt the language. So if you had learnt the language in the house from growing up, it was all you heard if you were growing up. Things would be very different if you learnt the language when you were a bit older. So if you started learning French or something at school, then the areas of the brain that are active when you are learning those languages and speaking that language will be very different from where your native language, the language you learnt as you were from birth, was there. So there are different brain areas that are active and responsible for language, and that's why it can be hard to pick up a language when you get a bit older. Because you've got to use very different brain areas than you do when you learn a language, from birth. So it's a really brilliant skill that if you've learnt languages really young from your parents or around the house, then you'll really retain that, and it'll be much stronger.
There's new questions coming in here. One relates to language actually. If you're deaf, which language do you think in? So I think that will really depend exactly on the case. So some people are deaf but have previously been able to hear, so that's different. They will probably be thinking in the language that they were used to or familiar with, but some people cannot hear from birth, and so they probably do not have a language to process, but they will learn sign language, many of them. And so parts of the brain that are normally important for hearing will then take some of the responsibility for learning sign language, and that's some way we can help them as well. Well,
I think we've got time possibly just for one more question, I think, because we're just about to wrap up the time there. So how about anyone that you like there? How about
how does the brain react when you smell? Well, that's another. You really like the senses, don't you? Well, I don't study smell, but many people do, and there is a region that is important for what we smell. But I really want to maybe end on a part of the motor cortex, which I really like. So one thing that, in case you don't know it yet, which I find really cool, is that when you wave your right hand, which we will do in a minute, you actually need your left motor cortex. So it's actually one of those sensory systems where processing crosses, which I think is always really intriguing to think about. That's how cool our brains are wired. But I think with that, we probably do need to use our left motor cortex and wave at you to say goodbye. But we really do love all the questions that are coming through, and keep thinking about your brain and how it works. Keep asking your teachers, keep inspiring to learn new things, and maybe one day you'll become a scientist yourselves.
Brilliant. Well, thank you. And I hope you've enjoyed it today. Thank you for joining. Good to see you.
And thanks to Barbara in the scanner, and thank you to all your brilliant ideas. Bye.